All right, today we're diving deep into one of the most incredible stories in all of biology. We're going to look at the step-by-step playbook for how a virus takes over a human cell. It's a wild tale of microscopic pirates, molecular machines, and all-out biological warfare. So you really have to wonder, right? How can this tiny little particle, so small it makes a grain of sand look like a mountain, hijack our bodies? I mean, this thing isn't even really alive on its own, yet it can commandeer the super complex machinery of our cells and turn them into virus-making factories. It's really a story of biological espionage. The best way to think about it is like a heist. Seriously. The virus has one mission and one mission only: get in, make copies of itself, and get out, all while dodging the body's security systems. Every single step is just a masterclass in biological engineering. Okay, so first things first, let's meet our microscopic burglar, the virion. That's the name for the complete infectious virus particle when it's just floating around. Outside of a cell, it's basically inert. Think of it like a seed just waiting for the right soil. It's what's called an obligate intracellular parasite, which is just a fancy way of saying it absolutely cannot reproduce without hijacking a living cell. It's helpless on its own. So once a virion gets into the body, its first challenge isn't just finding any cell. It has to find the right kind of cell. This is target acquisition, and believe me, it is incredibly specific. You know, viruses can't just phase through our skin. They need a way in, a portal of entry. So whether they're breathed into our lungs, swallowed with our food, or maybe they slip through a little cut in our skin, each route presents them with a totally different set of cells to try and infect first. And once it's inside, the virus isn't just randomly bumping into things, hoping for the best. No way. This is where the brilliant lock and key mechanism comes into play. The virus has these proteins on its surface. Those are the keys. And they are shaped to fit perfectly onto specific receptor proteins on a host cell. Those are the locks. It's exactly why the flu virus, with its specific keys, goes for your lung cells and not, say, your liver cells. The locks just don't match. And there's a great scientific term for this specificity, this pickiness. It's called tropism. It's just the virus's preference for a certain cell type, a certain tissue, or even a whole species. And it's all decided by whether a cell has the right molecular locks on its surface for that virus to grab onto. Okay, so our virus has found its target and it's latched on tight. Now comes the most critical phase, the breach. It has to let its precious cargo, its genetic code, past the cell's main defense, that outer membrane. And they've pretty much got two main ways of pulling this off. Some viruses, like HIV, use what's called direct fusion. Their outer shell just kind of merges with the cell's membrane and dumps its contents right inside. Simple. But others, like the flu, use a much more cunning trick called endocytosis. They basically trick the cell into eating them, wrapping them up in this little bubble called an endosome. It is the ultimate Trojan horse strategy. And influenza's escape from that Trojan horse? Oh, it is just brilliant. So once it's inside that bubble, the cell naturally starts making the inside more acidic. And that drop in acidity, the lower pH, that's the trigger. It causes one of influenza's key surface proteins to radically change its shape. It basically springs open, shoots out a harpoon, and forces the virus's membrane to fuse with the bubble's membrane. And just like that, the viral genes are free inside the cell. It's an absolutely incredible little molecular machine. So the viral genome is in. The breach was a success. Now the real takeover begins. The virus's one and only goal at this point is to seize control of the cell's production lines and turn it into a full-blown factory for making more viruses. But before it can do anything else, the virus has to unpack its instructions. This step is called uncoding. The protective protein shell, or capsid, gets stripped away, and the viral genome, its genetic blueprint, is finally released and ready to get to work.
But you know, this is where it gets a little more complicated. Viral blueprints come in all sorts of different formats. You've got DNA viruses, RNA viruses, and more. So how does each different type convince the cell to copy its specific kind of instructions?
Well, this table lays out the different strategies perfectly. So DNA viruses like herpes virus, they're pretty straightforward. They usually head to the cell's nucleus and just use our own DNA copying machines. But RNA viruses, like coronavirus or influenza, they often do their work out in the main part of the cell, the cytoplasm, and they have to bring their own special enzyme to get the job done. And then Then you have the masters of infiltration, retroviruses like HIV. They use this amazing enzyme called reverse transcriptase to turn their RNA code back into DNA, which they then permanently stitch right into our own chromosomes. So at this point, the cell's machinery is fully hijacked. It is reading the viral blueprints and it is churning out viral parts. The next stage is all about mass production, and just as important, planning the escape. Yeah, the cell is no longer working for itself at all. Its own machinery is furiously building viral proteins and copying the viral genome over and over. And these new parts often get herded into specific areas of the cell, which we call viral factories, where they get assembled into thousands of brand new virions. But how do these thousands of new viruses actually get out? Well, there are two main strategies here. Some, like HIV and influenza, use a stealthy approach called budding. They literally push their way out through the cell membrane, cloaking themselves in a piece of it as they leave. This doesn't kill the cell right away. The other method? It's called lysis, and this is the brute force approach. The cell is literally forced to burst open, releasing all the new virions in one huge destructive wave. And to buy themselves time to do all this, viruses have evolved some truly jaw-dropping tricks. See, our cells normally have an alarm system. They constantly show little pieces of what's going on inside them on their surface, like a status report for our immune system. But what you're seeing here is pure sabotage. The herpes virus makes a protein whose only job is to jam the transporter that puts those little pieces on the surface. By doing this, it makes an infected cell look totally healthy and normal, basically invisible to our immune patrols. The new virions are out. The host cell has either been destroyed or it's been turned into a long-term zombie factory. This leads to the fallout for us, the host, and it sets the stage for the next wave of infection. And you know, not all infections look the same. What's called a lytic infection is like active warfare. The virus replicates, it bursts out, and it causes an acute illness, like the flu. But some viruses play a much longer game with a latent infection. Here, the viral DNA goes into deep cover, hiding out, dormant, inside our cells. Herpes viruses are the masters of this. The virus that causes cold sores can just hide out in your nerve cells for years, waiting for just the right moment to wake up and reactivate. But for the virus to truly succeed, for it to survive as a species, it has to spread to a new person. And this final step in the grand plan is called shedding. So whether it's through a cough, or a sneeze, or some other way, the newly made virions are released back out into the world, ready to find a new host and start this whole incredible process all over again. And just like that, the cycle is complete. From one single inert particle that just happened to find the right lock on the right cell, an entire army of clones has been produced, been released, and is now ready to launch its own invasion. And really, that brings us to the final thought here. This isn't some static process that never changes. It is a microscopic arms race that has been waged for millions of years. Every single time we develop a new vaccine or a new antiviral drug that targets one of these steps, we are putting immense evolutionary pressure on viruses to find a new way around it. So the question isn't if they'll adapt, it's how.